I'm Matthew.
I'm Michelle.
I'm Minnie.
I'm Paige. I'm Denise.
I'm Dan.
I'm Sarah.
And I'm Jessica.
My name's Michelle. I'm a fish biologist at the NOAA Fisheries Lab here in Panama City, Florida.
And I'm Matthew, and I'm an ocean engineer at Naval Surface Warfare Center, Panama City Division. In today's episode, we will talk about the technologies used to help track the movement of different marine species and to understand migration patterns. We'll look specifically at the movement of sharks. By examining distribution and migration patterns, biologists can understand the impact of things like habitat loss, overfishing, and natural disasters.
We'll explore the external anatomy of a shark and examine the sensory organs used by sharks to navigate long distances, to find spawning grounds and food. We'll also take a closer look at the differences in the shapes of sharks' teeth.
We will introduce you to remotely operated vehicles, or ROVs, that are used to perform various operations underwater.
We'll learn about water quality parameters and how those parameters impact the distribution and movement of sharks.
And then finally, we'll dissect and discuss the internal anatomy of a shark.
We're excited to share this segment with you, so stay tuned for Side Notes
Marine Science.
So how do we know where or when sharks move? Do all species exhibit the same behavior? Do they move at the same time? Or do they go to the same location? To better understand movement of animals, biologists use trackers to analyze all factors that may influence migration.
NOAA has collected data on over 20,000 migrating sharks in the Gulf of Mexico for over 25 years, tagging sharks each year with the goal of recapturing them months or years later to measure how much they've grown and where they are.
Elasmobranchs are a subclass of fish composed of three families: sharks, skates, and rays. Instead of bone, elasmobranchs have skeletons made of cartilage. Sharks have 5 to 7 gill slits, which help to extract oxygen from the water as sharks continuously move. Movement patterns of elasmobranchs helps biologists understand how many sharks are in a given area and when those sharks arrive and depart.
There are different types of trackers that we can use on animals. Some let us know where they are at when they come to the surface using satellites. Satellite trackers are great because they can work all over the world, but the animal has to come to the surface for us to know where they are.
Information on swimming depths, water temperature, and a daily record of geolocations can be stored and downloaded from pop-up archival transmitters, also known as PAT tags. These satellite PAT tags can collect and relay data for up to 12 months, depending on data recording intervals.
The second type of tag is the acoustic tag, which provides data on fine-scale movements within a boundary of stationary receivers, which collect data when a shark swims within half a mile of the receiver. Several groups in a research community work together to share information from tag data collected on their network of receivers.
With these tracking tags, we're able to follow these sharks during daily and annual migration. Migration is essential for the survival of many marine animals, including sharks, and occurs during various times of the year and at particular periods in an animal's life cycle. Sharks migrate because of temperature, feeding, spawning, and avoidance of natural or human-made disasters. Migration for food happens on a daily basis and usually coincides with the migration pattern of their prey. Sharks typically consume squid, fish, and even turtles. Knowing what sharks eat is critical to protecting their food source, which affects their growth rates and lifespan.
Migrating marine animals travel long distances and often encounter marine debris. Marine debris is floating or submerged trash that can be ingested or entangle the animals, restricting their movement, growth, and digestion. Tracking sharks allows us to know their proximity to areas of high human activity, like developed shorelines and fishing areas, which could increase their probability of ingesting plastics or getting entangled in fishing gear.
It's important to be able to track marine life so that we can understand how humans affect their migration patterns. It's our responsibility to respect these creatures' homes and cohabitate with them peacefully.
Hi, I'm Dr. Andrea Kretz, and I am a fisheries biologist with NOAA in the Panama City Lab, and I research endangered small-tooth sawfish, as well as investigate the trophic ecology of sharks and rays and teleost in the Gulf of Mexico. So small-tooth sawfish were listed on the Endangered Species Act in 2003, and over the last century or so, the population of small-tooth sawfish has greatly declined, and this is primarily due to interactions with humans. So this right here, this is the rostrum of a sawfish, and you can see they have all of these very sharp and large rostral teeth. So as you can see with these teeth, you can imagine that these teeth can be very easily entangled within a net, and it'd be very difficult for sawfish to free themselves. So unfortunately, if they get caught in a trawling net or any other type of fishing gear where they can become entangled, it's most likely that these sawfish are not going to survive that event. Since the start of our survey in South Florida in Everglades National Park, which was established in 2009, we have tagged over 350 juvenile small-tooth sawfish. In addition, we have tagged over 25 juvenile small-tooth sawfish with these internal acoustic tags, which can last for up to between 4 and 5 years, which can greatly provide us information about habitat use and movement ecology throughout the ontogeny. So this right here, this is a VR2 receiver. This can detect the acoustic signal given off by these acoustic tags right here, which we internally implant in small-tooth sawfish. Now these acoustic tags are uniquely coded with a unique identification number, and when a sawfish that is tagged with one of these acoustic tags swims in relative close proximity to these VR2 receivers, this receiver can record and archive that unique fish ID and the date and timestamp. And then scientists can go back into the field at a later date, and we can download this data, and then we can get information as to where these sawfish are, specific dates and times, and then we can get an idea of their movement patterns. So through our tagging program, we have learned a great deal more about the movement ecology of juvenile small-tooth sawfish. Previously, we knew that sawfish would stay within their nursery habitat within their first 1 or 2 years of life. With the addition of these acoustic tags, we have learned that these sawfish are actually moving greater distances at a younger age. So sawfish approximately 4 to 5 feet in length are actually leaving their nursery areas. It's a bit earlier than what we had hypothesized, and they're moving longer distances. We've actually had some sawfish at about 5 foot, 6 foot in length that we tagged in the northern part of Everglades National Park that have traveled all the way down to the Florida Keys, which is phenomenal and information that we did not have prior to the technology of acoustic tags. With marine life being underwater, it's hard for us to easily observe the movement patterns of these animals, but with the tracking tags, we are able to better visualize and understand when and where they move. All of this data will help us to protect the remaining population, reduce threats, and protect habitats.
Sharks migrate long distances during certain times of the year. On a daily basis, they migrate short distances to find food. So how do we know where sharks are going, Matt?
Well, in this segment, we're going to look at the ampullae of Lorenzini, a sensory organ that helps sharks detect magnetic and electromagnetic fields and helps them navigate and find their way back home.
So here we have a juvenile blacktip shark that we'll use to discuss the external anatomy, and later on we'll do a dissection to better understand the shark's internal anatomy. Before we get to the sensory organs, let's look at the shark's skin. The skin of a shark is quite unique. It's actually covered with small teeth-like scales called denticles. This helps protect the shark from injury and reduces friction during swimming. Speaking of teeth, sharks have several rows of teeth that they continuously lose hundreds of teeth over their lifespan. Did you know you can identify the species of a shark by its tooth shape? Let's take a look at the tiger shark. As you can see, it's got curved teeth in comparison to a great white shark. Questioning tooth shape actually allows biologists to understand differences in the prey sharks consume. Because of their serrated shaped teeth, tiger sharks consume more turtles compared to sharks because of this difference in tooth shape. The shark biologists at the Panama City Lab often find whole turtles in the stomach of tiger sharks. So to sneak up on their prey, sharks use the color of their skin to camouflage themselves. This decreases their visibility from above and below, which is actually called countershading. So this is why sharks are light on the ventral side, which is bottom, and dark on the top, which is called the dorsal side.
Besides camouflage, sharks use other techniques to find their food. They use the typical senses, sight, smell, hearing, or vibration, but they also have this own unique sense that detects electricity. On the snout of the shark are tiny pores called the ampullae of Lorenzini, which are filled with a gel-like substance that helps detect the electricity present in fish. So in this video, researchers at Florida Atlantic University and Dr. Stephen Kajura are looking to see how well a bonnethead shark can use just that electric sense from the ampullae of Lorenzini to locate a simulated prey item. So just like fish put off an electric field, we simulated one using electrodes and hid it below the sand so that it can only use that sense to localize on this object. And as the shark swims around, it goes and bites towards those electrodes, and that gives us an idea of how quickly the shark can turn and how quickly it can cue in on that electric field sensation. As you can see, sharks are very intelligent creatures, and with the help of their external organs, they can accurately navigate the waters to find food, they have the camouflage to hunt, and use their razor-sharp teeth to eat their prey.
Stick around, because later on, we'll dissect a shark to learn more about its internal organs. So welcome back to Some Fin Special. So now you know sharks can see, smell, and hear. But how good is their hearing? Can you guess what the farthest distance a shark can hear? Is it A, 4 feet, B, 85 feet, C, 485 feet, or D, 850 feet? If you chose D, 850 feet, you're correct. This is more than 2 football fields combined. The acoustico-lateralis system is the combined hearing structure and lateral line which helps sharks absorb sound from all directions to orient their balance. Tiny hairs, or acoustico-lateralis, along that lateral line detect vibrations of prey movement through sound. This advanced hearing adaptation is one of the reasons why sharks are the apex predator of the ocean.
To understand ocean engineering and underwater navigation, we'll learn about the many ways that NSWC Panama City and NOAA use remotely operated vehicles, ROVs, to collect underwater data.
ROVs are commonly used by the Navy and NOAA because they're advanced vehicles that allows for a safe retrieval of underwater data. And these vehicles can be outfitted with specialized tools, such as cameras and acoustic equipment, that give us an insight into the underwater world that we wouldn't be able to see or hear ourselves.
ROVs can be really small and be used to look underwater where we may not be able to get to. This is really helpful if the water is really deep. They can also show us what is down there by streaming video or sonar feeds up to engineers and scientists who can sit above the surface of the water. ROVs might also be really big. The cables that run full lengths of the ocean are laid and buried by ROVs. They can also inspect really large structures underwater, and they have attachments to do jobs down there, like drill holes or grab onto something that was dropped on the ocean floor. There's 3 axes that our ROV can move on. It can move forward and back, and we'll call that the x-axis. It can move side to side, and we'd call that the y-axis. And it could move up and down, and we could call that the z-axis. It could also rotate about any of those axes. So it could roll side to side, it could yaw back and forth, or it could pitch up and down. If we could move and rotate about all of these axes, we would say this ROV has 6 degrees of freedom.
So how do you control up and down or forwards and backwards? Is it similar to driving a car, just with the added ability to go up and down?
Up and down is controlled by this motor in the middle. Going one direction moves the ROV upwards, and moving the other direction moves it down. The 2 thrusters on the side control which way we turn and whether we go forwards or backwards. Moving the right thruster turns us to the left, and the left thruster turns us to the right. If we use both of them, we can move forward or backwards. Controlling one thruster to turn a different direction is called differential thrust. If the thrusters were placed too close to the top of the ROV, it might always face downwards. And if the motor in the middle is placed off-center, then the ROV might tilt as it moved up and down.
So if the ROV is too dense or not dense enough, what would happen?
If the ROV is too dense, we might sit on the bottom and not get enough thrust from the thruster in the middle to ever pull us off. If the ROV is not dense enough, then we might sit on the surface the whole time and never get to explore the big world underneath the ocean's surface.
So is this ROV powered by a battery, and does the battery size affect the ROV in some way?
In this ROV, we run a cable to a battery over here. But ROVs on a boat may choose to run a cable up to a generator on board the boat to stay powered for long periods of time. There's other underwater vehicles that don't use cables to batteries, and they have a battery on board. Those batteries can often be heavy and take up a large portion of the space and weight of the entire vehicle. So, Michelle, let's put this in the water and see what it can do. We have a right thruster control to move forward and back, a left thruster control to move that thruster forward and back, and then a button for both up and down. This system is built with just PVC pipe, and you can add foam or take it away to make it the right buoyancy, and students can use these and compete with each other to do different tasks. So maybe they are underwater together and pick up something and attach another piece of PVC on the front with the little hook to go get rings at the bottom, just like you would in a swimming pool. And it gives some good experience with both learning how to control a system and learning how to design something to do a certain task.
So if you're in school, ask if there are opportunities to learn more about making robotics. Experimenting with vehicles is a huge help in learning about what careers in STEM may look like, and you can get a jump start on learning about interesting topics like controlling underwater vehicles.
Yeah, my name is Chris Gardner. I'm a fisheries biologist with NOAA in Panama City. I do underwater video surveys. So ROVs come in all shapes and sizes, from the small sea perch models. However, we have some that are the size of cars that can go down thousands of meters underwater. So this ROV is controlled from the surface. There's a big tether that goes all the way up where it gets power, and it sends video up to a monitor. It's equipped with 2 cameras in the front so we can rotate up and down. Has lights, the lasers are up front. So scaling lasers are just 2 lasers that are parallel, and ours are set at 50 millimeters apart, so about 2 inches. So if we see 2 dots on a fish, we know that that's about 2 inches in real life, so we're able to kind of estimate out how big the fish is. You basically play it just like a really expensive video game. You have lights, you have thrusters, which are propellers that go back and forth. Yeah, the most difficult part is managing the tether and the current. So we have this thing, in this one we have 100 meters of cable, which is over 300 feet. So that's longer than a football field of cable that can be out in the current and drag this little machine around. So with ROVs, anytime you take a sophisticated piece of electronics and put it in salt water, you might have some mishaps. So we spend a lot of our time maintaining, replacing circuit boards, and really troubleshooting these things, because even though they're small, there's a lot going on in here. If you're interested in ROVs and marine biology, I highly encourage you to visit noaa.gov to learn more about current research with ROVs.
The quality of water is vital to the health of our marine species, and water parameters such as salinity, temperature, and oxygen all affect the distribution and migration of sharks.
The first parameter that affects movement is salinity, which refers to the amount of salt dissolved in the body of water. We're surrounded by salt water that varies in concentration depending on the proximity to freshwater sources and the amount of precipitation. Salinity is measured in parts per thousand, with oceanic water having the highest concentration of salt compared to bays, mangroves, or estuaries, which have freshwater influences from rivers and rainwater runoff. So we've collected water from the St. Andrews Bay right off of the FSU PC campus. And in this sample, you can see the hydrometer goes from 0 to 40, meaning 40 is the highest salinity you can get, and 0 being no salinity, which is fresh water. So our water sample is coming in at 27 parts per thousand. So typically in the ocean, you have between 30 to 35 parts per thousand. So we're pretty close to the Gulf of Mexico, so our sample is reading higher in salinity, but if we go further north, near Deer Point, our sample would have less salinity, so it would be closer to the 5 and below to 0.
So how does salinity affect migration in sharks?
Well, sharks typically hang out in salt water and avoid fresh water, except for the bull shark. The bull shark will migrate into freshwater rivers to search for prey.
The second parameter which affects migration is water temperature. A variety of species, including some humans, move seasonally to warmer areas. Regional water temperatures and water depth temperatures distinguish where sharks live and migrate. The sharks are found in 3 different temperature regions: polar, less than 41 degrees, temperate, 41 to 70 degrees, and tropical, higher than 70 degrees Fahrenheit. Also, sharks can be located at 3 different depths: shallow, pelagic, and deep water. The bull shark, along with many species of sharks, live in the temperate region and remain in shallow coastal waters.
Oxygen is essential to the survival of every living organism. Oxygen is produced by aquatic plants like kelp, algae, seagrass, and even coral. Dissolved oxygen is measured in parts per million, with a suitable water having 5 to 6 parts per million.
Are there areas in the ocean that are not suitable for life because there is no oxygen?
Sadly, in the Gulf of Mexico, near the mouth of the Mississippi River, there's a large area called the dead zone. This area appears seasonally and can reach the size of the state of Massachusetts and cannot support marine life because of the high concentrations of nitrogen levels, which are coming from the industrial and agricultural pollutions and oil spills. The dead zone continues to grow each year and impact marine organisms severely.
By analyzing shark migration patterns, biologists can determine temperature, salinity, and oxygen preferences for each species and how certain areas are used at different times of the year, and which areas are avoided in the case of the dead zone.
So sawfish are in the same group of family as sharks and rays, which are elasmobranchs, and sawfish are actually rays. So they're cousins of sharks in that they have a skeletal system that is comprised of hardened cartilage. Sawfish are different from sharks also in that they are dorsoventrally compressed, which means they are top to bottom compressed. And one main difference between a shark and a ray, such as a sawfish, is the placement of gills and their mouths. So sawfish have their mouths on the ventral or underside as well as their gills, whereas sharks have their mouths on the front of their heads as well as their gills are located on the sides of their bodies.
Before we begin this segment, we'd just like to warn you that we will be dissecting a shark. So if you do not like the sight of blood or get a little squeamish, please skip this part of the video. So sharks actually regulate their buoyancy much different than a fish. Sharks do not have swim bladders like fish do, and they actually control their buoyancy up and down in the water column with their sizable livers. So what we're going to do is open the shark up to see the internal organs and actually take a look at that liver and how big it is. Matt, do you actually know how to tell the difference between a male and a female shark?
The claspers right in
Absolutely.
here?
So for male sharks only, they have extra 2 fins right on their pectoral fins. It's called the claspers. Only males have this fin, and that's how you can tell the difference between a male and female shark, if you ever get that close to a shark. So as we cut towards the head of the shark, I'm cutting down past the muscle of the shark. And as you can see, the first thing you see is the liver. So these 2 lobes right here are the liver of the shark. And they're filled with that squalene oil, which allows that shark, in addition to its cartilage, to float up and down in the water column. So inside of the shark, it's pretty basic internal anatomies. They share similar organs to us in that they have a heart, kidneys, a stomach, and intestines, but no lungs because they have gill slits. This shark actually has something in its stomach. So we could open that stomach up. Ooh, what is that? That is probably a fish. Inside shark stomachs, they have typically a larger level of acid because they cannot cut up their food with a fork and knife like we do and chew it, so they ingest their prey whole. So they will digest it quicker with the amount of acid. So typically, if we can get the shark and open it up, we can find the remnants of that species of fish or shrimp to tell what species it ate. And actually what sticks around is the otolith. So in all fish, there's a tiny little bone. It's actually 4 of them that stick around. And as biologists at the lab, they can actually go through those bones and tell which type of fish that the shark ate. So it's a bit of detective work in determining what that shark ate.
Sharks consume a variety of prey, which they most often eat whole. Sharks have large stomachs and digest their prey efficiently, using a spiral valve intestine system.
As we go further up, we'll get closer to the shark's heart. And as you can hear, the cartilage, as I'm cutting it, it's kind of like a sternum in us. Sharks have also that cartilaginous sternum. And there is our shark heart. Is that good? So their heart is protected just like our heart is protected with a sternum, but their sternum is made of cartilage. So it's pretty hard and it protects that delicate organ. So as you can see, inside of a shark is pretty similar to us humans, where we share a heart, a liver, also stomach and intestines. Sharks are pretty cool because they are digesting really quickly. But as biologists, we have to determine what they ate so we can actually figure out where they're staying, where they're migrating to, and what prey items they're consuming.
Biologists have learned a lot of information from doing dissections or necropsies to better understand differences in diet, reproduction, and lifespan for each species of shark. Taking tissue and organ samples helps pathologists determine if disease, chemicals, or other issues were the cause of death. This information helps biologists make informed decisions on the management of species and their habitat.
If you encounter a shark that has a tag like this on the dorsal fin, please measure the shark's length from the snout to the fork of the tail, record the tag number, the date, time, and location of capture, and contact NOAA Fisheries Panama City Lab at 850-234-6541.
The ocean is full of life, and it's a beautiful world that we don't often get to see. With the help of new technology, grants, and general interest in the ocean, we're gaining new information and research about marine life.
The ocean needs our help. With pollution, overfishing, and disasters increasing, the health of our oceans may continue to decline.
The good news is that we all can do our part to help reduce our impact on our oceans by following some sustainable practices. Awareness is key, and making small changes in our behavior can make a huge difference in the health of our planet. One change is reducing our dependency on single-use plastics and purchasing reusable products like shopping totes, metal straws, and reusable water bottles. Another opportunity to help is by volunteering to pick up trash on our beaches to help remove plastic that could be ingested by turtles, birds, or sharks.
If you are interested in marine science or want to know more about what we have discussed, please check out the links provided, where you will find out about ways to get involved in your local community. I'm Matthew.
And I'm Michelle. Thanks again for tuning in to Side Notes Marine Science. We hope that you're able to get out in the water soon and explore our beautiful oceans. So, red.
What does sharks eat? Let's start right there.
Am I lying?
And some turtles.